Hi, today I am going to present quite basic thing, but which is quite demanding for most of the biologists who are doing some kind of research or basic studies and using some bigger data sets or larger number of genes to analyze their patterns in different perspective and to correlate them with the phenotypes or biological functions. So this is quite basic thing, but I have experiences that most of the people loses the basic idea to understand the basics even. For example, what is p-value and how to calculate the p-values, what is the fold change, and what does it mean when we are talking for two-fold changes, 2.5 fold changes, and what are the interpretations. So I selected this topic because this is quite common and trivial to understand. Many people knows how to calculate the fold changes, p-values, differential expressed genes, but they do not know the basic idea. So I decided to proceed and introduce the basics of fold changes, p-values, and many more factors for differentially expressed genes or differentially expressed proteins or something else. Now, so what do you mean by differentially expressed genes or proteins? Differential expressed gene profiling is a computational and experimental method used to identify with statistically significant changes in expression levels or patterns between two or more groups or conditions, such as disease versus healthy tissues or cells before and after the treatment. It involves normalizing expression data, performing statistical tests to find the genes that are up or down regulated, and it analyzing the results to understand the biological mechanisms, discovers the potential biomarkers, or identify the therapeutic drug targets. This is one of the example to understand what I told in the text here. So for example, here you can see the data sets as case 1, 2, and 3. So genes and then their cases, control, cases, control, cases, control. So here you can see the intersection of the genes in terms of fold changes in different case and in different controls. And again here you can also sort them based on ascending or descending orders. Now, so you need to basically understand the concept behind differentially expressed genes or proteins or differentially expressed microRNAs or long non-coding RNAs. So please also don't forget to subscribe our new channel also, that is Change by Science, and another channel, old one, is Bioinformatics for All. These two channels contains different things. For example, Change by Science will have the longer videos in details, and mostly it will include the technicals of the tools and techniques used in biosciences or biological sciences or medical sciences. Now, so the key steps and methods behind differentially expressed genes prediction are, for example, data integration, collection, and integration. So gene expression is typically measured by using methods like RNA sequencing, microRNAs, and RNA sequence provides a more detailed snapshots of the transcriptome. Now it comes to the normalization. So these are the pre-process before the fold changes or differentially expressed gene calculation. You need to go for normalization. So the raw data or expression data is normalized to account for technical variations such as differences in sequencing depth to ensure comparability between the samples. Now, statistical analysis. This is quite important and it is very useful to understand. Statistical tests are performed to determine which genes are showing statistically significant change in the expression pattern. This is crucial to distinguish the real biological differences between random variations. Commonly used methods include the tools like feature counts, DESEC2, AGE-R, which often use statistical models like the binomial distributions. But you can also write your own code, and it is quite simple if you understand the fold change, p-values, and how it is calculated. Then, for comparing more than two groups, tests like ANOVA can be used, followed by the post hoc tests to identify which groups differ. So, here you can see the next point comes is interpretation of the different factors in terms of statistical analysis. So the results are interpreted to understand the biological significance of the differentially expressed genes. And first one is the log2 fold change. A positive log2 fold change indicates a gene is upregulated, while a negative fold change indicates it is downregulated gene. Now it comes to the p-values adjustment. Multiple testing correction is applied to adjust the p-values, controlling for false positives. Now, next point comes to the visualization or presentation. Results are often visualized by using volcano plots, which shows the fold change versus statistical significance, that means p-values, or MA plots. It is also showing the log2 fold change versus mean normalized counts. Now, after you calculate the fold changes p-values of the genes differentially expressed, then you proceed for the pathway analysis or biological function relevance. For further analysis, gene ontology or KEGG pathway analysis is performed to see which biological processes or pathways are affected as a result of the respective differentially expressed genes. And if I am saying the p-values adjustment, for example, this is the raw p-values and this is the corrected p-values. There is quite difference between raw p-values and corrected p-values. I will also explain the difference between raw p-values and FDRs, that is false discovery rates. Then how and where you can implement these things? You can implement these fold changes, p-values, or visualization or pathway analysis for proteomics data, microarray data, RNA-seq, scRNA-seq data also. So basically for all kinds of multiomics data for all the species, you can perform such analysis. Now this is the representation to show the gene ontology and biological function analysis. So this is clearly showing what are the pathways showing highly enriched or gene ontology terms highly enriched, and what is the p-values and why we are saying the highly enriched terms. Now applications are diverse and quite common. So it can be applied for disease research, identifying the genes that are different between healthy and diseased samples to find the potential biomarkers for diagnosis or prognosis, or to understand the disease underlying mechanisms or features. Then it comes to the drug discovery, finding new drug targets by identifying the genes whose altered expression is linked to the disease. For example, a drug could be developed to inhibit a receptor that is highly expressed in case of cancer cells. Now it comes, third application is treatment response, evaluating the effectiveness of the treatments by observing how gene expression pattern changes in response to the therapy or drug treatment. Then the next application is environmental studies, understanding how organisms adapt to different conditions such as thermal stress by identifying the genes involved in the responses. So these are the quite common and useful applications of the fold changes, p-values, and more things. But there are diverse applications, and it is used almost every day in the lab or in the universities at college levels. Now, what are the factors which you need to understand? Those samples, number of samples, sample sets or groups, and how many samples are present in each group, then p-values, fold changes. So these things you need to understand clearly. For example, here you can see two different colors of the circles. So one, you can see the left side here, another the right side here, and they are basically two groups. But if you want to understand the significance, di- different between these two samples, then you need to understand how many samples were present in one group and another group. So on that basis, you will calculate the p-values. Then you will calculate the mean of the values. Then you will calculate the fold change. So this is how it is calculated. So if in one group, the mean expression value of the specific gene is 10, and in another disease condition or another target group, if the mean average value is 5, then the fold change will be 2. That means the gene is down regulated because in the target it is less than the normal. So this is how it works. But let's go in details. So if you are performing the t-test for the purpose to generate p-values, then you need to understand what kind of samples you have. Are you going to compare two sample t-test to evaluate the differentially expressed genes from two different experimental conditions or phenotypic conditions? So variables could be named dependent data or and independent data, which provides two matrices of gene expression values from two different experimental conditions in all the samples. Then calculate the p-values and t-scores for the gene expression values in the two matrices. So here, this is how you can write the code in MATLAB. And then you will get the immediate plot. If you write the exactly this code, that will be the normal quantile plot of t. So what are the significant points here you can see? And then quantile you can see. Then the diagonal you can also see here. So a p-value in a t-test is probability of obtaining a test statistics as extreme as or even more extreme than the one calculated from your sample data. So assuming the null hypothesis is true, a low p-value, typically lower than 0.05, indicates that the observed difference is unlikely to be due to random chance, leading you to reject the null hypothesis and conclude the difference is statistically significant. So null hypothesis, what the p-value tells you is about null hypothesis. The p-value is calculated under the assumption that the null hypothesis is correct. For example, null hypothesis might state that there is no difference between the means of two groups. Then you need to understand the probability. A p-value of 0.05 means there is a 5% chance of seeing the data you have or more extreme data if the null hypothesis were true. Evidence against the null hypothesis, a smaller p-value indicates stronger evidence against the null hypothesis. A larger p-value suggests the data is inconsistent with the null hypothesis. So how to interpret? Compare the two, see a significance level, a predetermined threshold called the significance level alpha or 0.05 before the test. The most common alpha is 0.05. So reject or fail to reject. If p-value is less than alpha, you can reject. This is how you can write the p-value the command in any R or Python or Perl or MATLAB. So the result is considered statistically significant when the p-value is less than equal to 0.05, suggesting a real difference between the group means. If p-value is greater than equal to alpha or greater than, if you are using here less than, then here you can say greater than equal to, or if you are saying here less than equal to, then here you need to say greater than. So if you have p-values greater than equal to alpha, you fail to reject the null hypothesis. The result is not statistically significant, meaning the observed difference could have occurred by random chance. Now, you can see the more plots in terms of histograms of the t-test results. For this purpose, you can perform this thing by writing some different codes and commands or functions. So you can use also the permutation concept. So here you can see that you can get, if you write this command to calculate the p-values and t-scores using 1000 permutation tests, then it will display you the histograms of t-score and p-value distributions, which is here. But if you go for 2000 bootstrap tests, then it will display you this kind of plot, which is slightly different than the above plot. So this is how you can do the evaluation of the calculated p-values and you can satisfy yourself. Now, it comes to the p-value adjustment. That is also called FDR calculation from the p-values. So adjust the p-values for the FDR when performing multiple statistical tests, such as comparing thousands of genes. You must correct the p-values to control for the false positives. And for this purpose, you can use the command mafdr function from the bioinformatics toolbox in MATLAB to estimate the positive false discovery rate or PFDR. So how you can write this command? You can write this command here, and p-value is p-vals is basically for the stored values which you have already calculated. Then here, to adjust the p-values for the FDR, the most common method is Benjamini-Hochberg procedure, which ranks the p-values from smallest to the largest and applies a formula to create adjusted p-values or q-values that control the expected proportion of false positives among significant findings. You can perform this using statistical softwares or manually by sorting the p-values. Now, what is the Benjamini-Hochberg procedure? This method is more powerful than the conservative Bonferroni correction and is widely used in large-scale studies. Sort the p-values. Arrange all your p-values in ascending order from smallest to the largest. Assign the ranks. Give each p-values a rank based on its position in the sorted list. Then apply this formula, or you can think about like this: FDR equals false positive upon all the significant results. So determine the significance where you can compare the adjusted p-values to your desired significance level. For example, alpha equals 0.05. All the hypotheses with an adjusted p-values less than or equal to the threshold are considered statistically significant. And this is one of the example to see the false discovery rate, which is the basically proportion of false positives among all the significant results, how they differ from the uncorrected p-values. Here it is FDR corrected p-values for 4 tests, FDR corrected p-values for 5 tests. They differ each other depending on how strict you want to be on this p-values or adjust statistical significance. You can choose your option. Now, how to calculate the fold change or log to fold change, from where this term comes, when you will be able to say the log to fold change or log to FC or just fold change. So if you have normalized the gene expression value, and used the log to a scale to present all the expression values, then all the fold changes calculated will be log to fold change. If you have not normalized properly and not used the log to a scale, you cannot say log to fold change, because I have seen a number of times throughout the conferences and presentations, irrespective of the countries, the people used to make such small or minor mistakes. But you need to be careful. So, what you need to understand, first thing you need to understand, if you are going to calculate without any software, just from by pen and paper, how to calculate the fault rate. First thing, if you have in the healthy samples five data, five samples, if you have healthy five samples and 10 the cancer samples, then what you need to do, for each gene you take the mean of all the five values in five samples for each gene, and then take the mean. Similarly, do the same thing in all the 10 samples of cancer data set. Then divide the bigger number, bigger values with smaller values. And if healthy samples are higher, then and the cancer samples are showing the mean values lower, that means the gene is down regulated in case of cancer, specific cancer. And when you have opposite, for example, in cancer sample you have higher gene expression values with respect to the healthy samples, then it means the genes are overexpressed or upregulated. So this is how you can do it. Same thing your software or small programs does. So the fold change represents the ratio of expression between two groups. It is often log transformed to provide a more symmetric representation of up and down regulation. Examples of log to fold change calculations are here. So for example, how to calculate the things I have already mentioned, you need to take the mean values and then you go for some kind of plots, because before you go to say whether it is up or down regulated, you need to see two parameters. One thing is p values. If the p values are less than equal to 0.05, or FDRs are less than equal to 0.05. If you are taking FDR, then you can implement FDR. If you are taking just p values, the uncorrected p values, you can also take it, and there is no mistake and nothing wrong. So then second thing comes fold change. You need to assign the threshold of fold change and p-values. If both the things are falling in your condition, then you can say the genes are up or down regulated genes. If only one is falling, then it is quite difficult to say, because, for example, for p-values are not significant, then you cannot say it, because insignificant values you cannot represent. So this is basically a volcano plot which is used to present the all the genes, calculated genes or predicted genes for their differentially expressed patterns. This is the p-values for all the upregulated genes, and this is the probe ID, because this is microarray data. And this is downregulated genes, and their respective p-values are here. So basically, right side in this box, they are upregulated genes. Left side in this box, they are downregulated genes. So the fold change for upregulated genes are positive. For example, -2, -3, -1.5, -1.8. Sorry, 2, 3, they're for upregulated genes. And for downregulated genes, they are in -1.5, -2, -2.3, -3.5, something more. And these are things are rejected because it is not only p values, but also you need to think the fold changes. So both the things needs to be in the limit. Here the log 2, log minus log 10 of p values are there. This is log 2 of fold change. So both the things are needs to be considered simultaneously, not just one factor. If someone is doing this, it is wrong. A volcano plot visualizes the results of differentially expressed genes by plotting statistical significance or p-values, and then you need to think the volcano plot, which is basically available for different perspective. For MA, plot also is available, but depending on different perspectives. What are the purpose, how you want to present your differential express genes, it all matters. But it is you who will decide the visualization style. So using the two matrices, the p-values calculated by MATTEST and the probe set IDs, column vector of the labels provided. Use MA volcano plot in MATLAB to create a significance versus gene expression ratios scatter plot of the microarray data from the two experimental conditions. Now, you need to think about volcano plot. You can also do this thing manually. So create a volcano plot manually by using standard MATLAB plotting functions, which gives you more control over the appearance and the methodology. And for manual volcano plot, this is the details. So you need to define the threshold of p-values cutoff, threshold of fold change cutoff, and then you go for a scatter plot where you can plot all the fold changes values and p-values simultaneously by using some kind of command called hold on. And then you can go for line references if you want to add line and fold changes threshold everything. And then for labeling, manual labeling, you can use such commands to label them up. Now, what are the key takeaways from this lecture? So p-values can be calculated for hypothesis testing using the functions like t-tests, 2 or mat test or CORR functions. Then for FDR, the MAFDR function could be used in MATLAB. For fold change, calculate the ratio of the means between your experimental groups, then apply log2 transformation. Then volcano plots can be done by using volcano plot function or a scatter function or some other manual approaches depending on your experience in coding. So these are the examples of the calculated p-values and corrected p-values. Then right side figure A and B is showing the sets of genes, a small number of genes, and their expression values by using some kind of PCR technique. And then also RNA-seq approach. So they have been compared together. So which one is giving what kind of expression values and what are the differences? So one thing what is clear in conclusion from figure A and B is if a gene is showing overexpressed, it can never be down regulated in RNA-seq or other techniques in just one sample or in the same sample set. The values could differ slightly. It is possible, as you can see here. So this is the example for volcano plot. It depends on the your choice of visualization, how you want to plot it for the visualization purpose. Now, the most common software used for high throughput data, NGS data, for differentially expressed genes are is DESeq2, which is a statistical analysis package of R used primarily for RNA-seq data to find differentially expressed genes by modeling count data with a negative binomial distribution. It normalizes gene counts, estimates syringe dispersion, estimates variance, fits a generalized linear model, and performs hypothesis testing using a wall test or likelihood ratio test. The analysis identifies genes with statistically significant differences in expression between experimental groups by applying multiple testing correction to p-values. So the key steps are data preparation, normalization, dispersion estimation, and GLM fitting or generalized linear model fitting, hypothesis testing, multiple testing correction, result interpretation. So DESEC2 key steps are data preparation, normalization, dispersion estimates, generalized linear model fitting, hypothesis testing, multiple testing correction, results interpretation. Why the people are using DESEC2? It is specifically designed for count data, accurately modeling the variability inherent in sequencing data. It handles complex experimental designs, including multiple groups or factors. The shrinkage of dispersion estimates provides more accurate results than methods that do not share information across the genes. It includes the tools for filtering low count genes and identifying outlier samples. One conclusive point here is that DESEC2 cannot be used for microarray data set. But if you want to still use it, you need to write some small modules in order to incorporate in DESEC2. And this is the workflow for DESEC2 utilization for calculation of the differentially expressed genes of in case of RNA-seq data or whole genome sequencing or whole exome sequencing data. So you can then think about once you calculate the differentially expressed genes, then you can think by using either DESEC2 or HR. You can think about sample clustering, hierarchical clustering, multidimensional scaling, principal component analysis, or TSNE. Or you can also think about the multi-species expression networks, cytoscape, EPIC, WGCNA. Then you can go for gene expression clustering, hierarchical k-means clustering, self-organization map, or afterward, you can move for functional term over representation or functional enrichment analysis. For example, gene ontology terms, interpro terms, TIGR, FAMS, and then finally, you can go for the David or get pathway analysis or Panther pathway analysis or reactome pathway analysis. This is depending on you, but the most common thing is get pathway enrichment analysis. There are the summarized presentation. I have collected this figure from one of the very beautifully presented paper in Nature Communication, where it shows if you are working on gene expression data and you are interested to analyze the gene expression patterns or profiles, then you need to think about the steps. For example, fold change and ranking the profiles, then ranking the profile, merging, for example, integrating them together, then go for some kind of functional relevance and better visualization plots in terms of conclusion